I'd like to begin by acknowledging the traditional custodians of country. The QMS does this and recognises throughout Australia the continuing connection to land, waters, fungi and communities. We pay our respect to Aboriginal and Torres Strait Islander cultures and to elders past, present and emerging. Thank you everyone for coming tonight. Fran is one of those people who is doing some meaningful work as a specialist, and she specialises in Marasmius. Unfortunately, we don't have a beautiful herbarium library full of specimens like we should. So Fran finds herself doing a lot of field work in order to get the resources, the fresh material that she needs to do work on Marasmius. And she's going to talk tonight about some of the work that she has done in the field about foraying in the far north. And so what I'll do now is hand over to Fran.
Okay, thanks. Okay, so tonight's presentation is Foraying in the Far North, and it's about our trip, a field trip up to Far North Queensland in February. And thanks for the opportunity to share with you some of the excitement and the discoveries of my trip up north. Firstly, though, I would like to pay my respects to the traditional owners of all the countries that we visited, and to thank them for their custodianship of the lands of the wet tropics for thousands of years. These are truly special places, and it's a privilege to walk in the tracks of the original dwellers. Field trips of this duration, next, and distance don't just happen, and we had some specific goals. For me, that was to collect fungi in the genus Marasmius for my study, but particularly to try and find specimens of the horsehair fungus, Marasmius crinis-equi, one of the few Marasmius in the world that was originally collected and described in Australia. My pre-trip research told me that it was collected in Rockingham Bay. That was in a paper published in 1880. And the specimen from that collection is in Kew Fungarium in the UK. However, confusingly, other papers from almost the same time suggested Dalrymple Creek. And if you look at the map, you can see that Dalrymple Creek is not too far from Rockingham Bay, but also from the Richmond River in New South Wales and the eastern Gippsland in Victoria. But there are no specimens from those southerly areas, so I was hoping that I could re-find it in the north. And we planned to maximise our time in the Rockingham Bay and Dalrymple Creek areas. So have a look at the map, and you can see the places marked in red are the ones that we visited. The wet tropics stretch from almost 500 kilometres from the south in Paluma right up to the Cape Tribulation in the north. My travelling companion, Teresa LaBelle, had a special interest in looking for agaricus species, particularly the non-urban, non-yellow stainer varieties. National park permits are always essential for doing any collection, and they took almost 6 months to obtain, but it included writing to all the traditional owner groups, and there were 10 of them for those parks, for their permission. I wrote to them and then I spoke to them all by phone, but only had written responses from 2 of the groups. And I think that just illustrates that it is a long, slow process communicating with traditional owners and working on a relationship. Choosing where to go was part serendipity, word of mouth, offers of hospitality, connection with people who knew their local parks, and our own knowledge of good fungal spots. Obvious time constraints, we did only have 3 weeks, and part deliberate planning to cover as many potential locations for Marasmius as possible. The timing was critical. It just had to be the wet season. Marasmius have very ephemeral fruit bodies. They're here one day in the rain and gone in 2 days' time. So luck was on our side as we scored a La Nina year with rain at least every other night or day. I made contact with all the members of QMS who live from Townsville North, and some of you are on Zoom tonight, and it's lovely to see your names there. I was blown away by the positive responses and the eagerness to share our forays, to lead some and to offer hospitality, which was incredibly generous. And the following slides are just a small selection of the fungi we found and the places we visited. Our team consisted of my husband, Bob, who's my chief support, my supervisors, Dr. Teresa LaBelle and John Durnley, who is here tonight, Dr. Matt Barrett from JCU, and a cast of dozens of others. Next. Our first collecting spot was at Paluma, which is 60 kilometres west of Townsville, 1,300 kilometres from home, and over 800 metres above sea level. Starting at the Little Crystal Creek Bridge on the road up the mountain. And almost my first collection there was of a Marasmius, Marasmius tenuissimus. This is the slide that shows the Marasmius tenuissimus to the right and the left of the bridge. And it is one of the unusual members of this genus because it doesn't have a stem. Our hosts at Paluma, Andy and Will Cairns, had been introduced to us by one of our QMS members, who's Betty Jacks, whom I will mention again later. And within walking distance of our accommodation in the rainforest, we found all the following specimens and many more. So we had a whole day at Paluma, and there were 4 keen new members from QMS who joined us for the day. Penelope and her partner George from Magnetic Island, Diane and her partner Ian from Townsville. And they were all very excited as newcomers, delighting in discovering and photographing and learning about fungi. And Teresa was in her element as a teacher. And these are just a few of the wide variety of fungi that we saw. Those of you who are familiar with the orange ping pong bats, the weedy species that occurs in southeast Queensland called Favolacea colossera, will be interested in this native species, Favolacea pustulosa. That's on the left-hand corner at the bottom. So after our walk in the rainforest, we came back to the house and wrote up the specimens. We took lab shots and showed our new friends the process of taking samples for DNA, spore printing and drying the collections. And we had already found a horsehair fungus, which fitted the description of Marasmius crinis-equi. Betsy, who's a very keen observer and a botanist, had told us that she'd seen it there in years past. So we were looking out for it and we were very excited to find it close to the place where we were staying. So I want to just pause for a moment now in showing you the variety of fungi to talk you through how and where to find horsehair fungi, including Marasmius crinis-equi. Firstly, you need to look in rainforest, often near streams where it's very humid, in the shade, and generally in the aerial fungi. Look for dead leaves or twigs that are caught in a tangle of black threads, the rhizomorphs. And for those of you who don't know that term, rhizomorphs are bundles of fungal threads enclosed in a tough melanized coating that prevents dehydration and enables the passage of nutrients and water through the cords. They attach to understory plants and even trunks of trees. The rhizomorphs are plentiful and long-lasting. They can last sometimes for years. The fruit bodies, however, are tiny, sparse, and quite ephemeral. There are many different species that produce rhizomorphs, so they're not unique to Marasmius, let alone Marasmius crinis-equi. The wet tropics are absolutely fabulous for lichens. We went out at night and looked at these with a black light, and they fluoresced most spectacularly. So if ever you have a chance to get hold of a little black light, there may even be one for sale here. It's worth going out at night time to see what fluoresces. You'll be amazed. Another unusual fungus is the geoglossum, which is the earth tongue, an ascomycete, and a phallus on the right lower hand there, which had no skirt on it. And whether that was because it was deformed or whether it's a different species, I'm not sure. It was really large. From Paluma, we drove north through Ingham and 40 kilometres inland to Abagari State Forest, which was the nearest campground to Dalrymple Creek. Remembering Dalrymple Creek was said to be where Marasmius crinis-equi was found in the 1800s. It had a beautiful, Abagari State Forest had a beautiful tiny patch of riverine rainforest and another small area of melaleuca and eucalypt forest with a completely different suite of fungi. This one here is related to the common leathery goblet, the Somatoderma elegans, which we see here commonly, but it's more ridged and it's laterally attached. It often grows on old dead lawyer vines, though I'm told it may grow on other palms as well. The rainforest there had been flooded only a couple of weeks prior to our visit, and there were piles of debris around with lots of logs and many decomposers on the logs and in the debris. This was one place where we did find Marasmius crinis-equi on leaf litter, though still in a tangle of rhizomorphs. And I want you to note that wherever I use the term Marasmius crinis-equi applied to our collections, it's a field identification and it has to be confirmed by microscopy and DNA yet. However, as well as the rotters in Abagari, we found truffle-like little tiny things, glomeromycota, which are arbuscular mycorrhizas. So they're attached and partnering with the rainforest trees. On the forest floor, we found them. And also with the palm seeds, the little blue Penicillium coccotripicola, which was described by our own QMS member, Dave Holdham. And as well as that, this beautiful slime mold in the Myxomycota, Arcuria species. The other vegetation type in the park was fringing melaleuca and eucalypt forest with swamp mahoganies and red gums. And there we found lots of ectomycorrhizal fungi, like this beautiful member of the Russulaceae. In the Lactarius group, it's now called Lactifluus afwirabara, which means like Wirabara, which had a velvety brown cap and brown stem and brown edges to the gills. Other mycorrhizals in this small area included a number of different boletes, some with gills like the Phyloporus in the top left and Boletellus, as well as russulas and chanterelles. The russula here was one of the green ones, Russula viridis group. As Dalrymple Creek had been recorded by Miles Berkeley in 1881 as one of the sites for Marasmius crinis-equi, we had to check it out. It was a beautiful creek with a very narrow strip of vegetation forest beside it. But it was surrounded by thousands of hectares of plantation pines and huge sugarcane farms. The forest near Dalrymple Creek was very disturbed and weedy and quite open. It may have once been suitable habitat for Marasmius crinis-equi, but is now much more suited for cattle. And I must say, though, that there is a walking track from Dalrymple Creek over the mountain range to the coast following an Aboriginal path, and that may pass through some rainforest, which is suitable habitat for fungi, but we didn't go that way. It's a difficult track and takes a couple of days. Instead, we headed to Cardwell and Rockingham Bay, which was named by Captain Cook in 1770. It's a large bay lying off Cardwell, north of the Hinchinbrook Channel, and it's 25 kilometres long and extends up to Tully Heads. It's a beautiful stretch of water. Cardwell, back in the 1850s and 60s, was going to become the main port for the newly opened up cattle properties of the north and the administration and financial centre for the area. However, it was quickly realised that the shallow water was quite treacherous, and for various other reasons, Townsville became the preferred port. Cardwell then drifted into the sleepy village that it remains, just a fish and chip stop en route to somewhere else. I came to realise that Rockingham Bay in the 1860s was shorthand for the whole district, a large quasi-administrative area for the coast, from the coastal range south of Cardwell to the Murray River in the north, including all the islands, the coastal plain, and the ranges behind, and even the settlements and properties west of Abergowrie. That district covers 5,000 square kilometres. The area had in fact been relatively closely settled by Aboriginal groups because of the rich resources in the sea, the rainforest, and the open forest between. The history of European settlement there is littered with bloody conflicts as the local people were displaced. But a number of Aboriginal groups still have traditional connection to the land there. However, today, apart from areas protected by national parks, much of the region is covered with pine plantations and various other farming pursuits. Fortunately for us, the national park system is quite extensive in this area. So on the first day we were in Cardwell, while I was still imagining that we might be finding Marasmius crinis-equi in or near mangroves, we visited the Edmund Kennedy section of Girramay National Park just north of Cardwell. We didn't see anything of my target species, but we did find some other very interesting fungi. This Marasmius was present in hundreds, maybe thousands, growing on the acacia phyllodes beside the road. And it's a relatively uncommon, in a relatively uncommon section of the genus. And the cap cells look to me like little hedgehogs. So that was interesting, but not the target. The next exciting find was this Pycnoporus puniceus on the left. It's fleshy with large irregular pores, and I've put up there to compare it with the Pycnoporus sanguineus with its flattish thin brackets and fine pores. According to Matt Barrett, this one of the Pycnoporus puniceus is only the third collection in Queensland, I think, maybe in Australia. So it's a rare one and a very interesting one. So while we fed the mosquitoes as we walked to Wreck Creek through the mangroves, we came on another different fungus. And this small, brightly coloured fungus was growing in dense clumps from almost buried wood. It was dead roots. And although there'd been a flood through there only the week before, so that may be why it was buried wood. But it appears that as the fruit bodies mature and die, the caps turn black and they sort of jellify and the stems remain, making this really tough bunch of caps and stems. At first, I thought it might be a Marasmius, but the jizz wasn't right. The cap and the stem texture were too smooth and slippery, and the habit was just unusual for Marasmius. And microscopy has confirmed that it is not Marasmius, but I have no clue as to what it really is. So that's one to be looked at in future. So having failed at the Edmund Kennedy Park, we next tried the scenic loop around Cardwell. And it takes in a number of small parks, lookouts, and popular waterholes. There are waterfalls and swimming holes in a number of places. Surprising to me was that the habitat was open eucalypt with grass trees and quite a number of wildflowers, almost like a mountain heath. The native hibiscus was one of the most stunning of the flowers. I love its name. It's called Abelmoschus moschatus, and it was a large flower and really beautiful. It's really well worth a stop in Cardwell simply to do that scenic loop and enjoy the swimming holes, the rock pools also fringed with the flowering golden penders. So it was not Marasmius country, but a number of other species, such as these boletes, some of which are known, like the Ostroboletus on the left, and many which are unknown. Nigel Fechner, who has studied boletes, his best guesses are Hymioporus for the intense yellow-pawed one, and possibly Gyroporus for the lower right one. But he said there were things that didn't quite fit. There were also beautiful corals in the area there, including the blue-tipped Phaeoclavulina and Ramarias of different species, and then other corals like the little Cytenopogon on the right. And it is a clavarioid one with very flattened branches and sharp points on the ends of the branches, and it has spores that are warty. So on our last day at Cardwell, we visited Murray Falls section of Girramay National Park, which is probably about 30 kilometres as the crow flies from Cardwell. It's 40 kilometres by road, and it included rainforest along the Murray River Valley below the falls. It's about 84 metres above sea level, so not terribly high. There'd been a little rain the day before and a very heavy and dramatic thunderstorm through the night. So the morning was steaming, the creeks were all roaring, and the rainforest was dripping. Perfect for fungi. It was just the morning to see cups and discs of the ascomycetes at their best. This was one of several places where we saw the gorgeous, hairy Cookina tricoloma, the second down on the right-hand side. The habitat was, as you can see, dense rainforest, and it was close to a little stream, and it was very thick. And there we found masses of Marasmius crinis-equi in the understory, often on lawyer vines, sometimes tangling several different plants, incorporating dead leaves and twigs, occasionally loosely wrapping around a small tree trunk from way above my head down to the ground. And it consisted of tough, moderately fine black threads with tiny 1 to 5 millimetre diameter white to buff caps. And they stood out like little white dots in the dark forest. Each cap is dimpled with a central dark spot and has 7 to 8 gills, meeting in a small collar around the hair-like stem, which generally arises from the rhizomorphs, although occasionally from small twigs. At last, a collection with enough fruit bodies that could form an epitype from the original type area. I was very excited. And then back in the picnic area, we found this beautiful, almost pink Philoporus species. Now, Phylloporus are just beautiful fungi. They're related to the bolets, and you can find one and you'll say, yes, this is Phylloporus, and immediately you say, but that's all we know about it. Somebody needs to take this on as a group to study, and maybe one of you people could do that. But even more importantly, Teresa found her first agaricus specimen, probably an undescribed one. It was a day to celebrate. So we did that by having a dip in the rock pools of Murray River. And it was nice to cool off, I must say. There were lots of specimens to process that night, and we were very grateful to be hosted by another fungi friend who works in biosecurity in Innisfail, Kerry Pearce. She loaned us her veranda for sorting, describing, and drying our collections, and sent us on our way next morning for a short foray at Josephine Falls at the foot of Mount Bartlefree before we headed to Cairns. Now, a visit to Cairns is not complete without a few hours at least in the Flecker Memorial Botanic Gardens. And whether you're interested in tropical plants or birds or butterflies or fungi, the gardens are a great place to visit. As well, one of our new QMS members, Bridget Gower, takes nature photography walking tours and night tours of the gardens. So she knows the place really well, and she knows where to find fungi, and was only too happy to take us for a walk. One of the most amazing non-fungal things to see there was a whole fence, meters and meters long, covered in Aristolochia vines, being munched by scores of Cairns birdwing butterfly larvae of all ages. Once they've got to the stage of pupating, they move on to a different plant, which is probably a good thing, as their food supply on the Aristolochia is being eaten away. The diversity of fungi growing basically in the garden mulch is quite extraordinary. The Climacodon was the size of a large cabbage, and it's in the polypore family, but it actually has teeth, not pores. And this one was fairly old and beginning to deteriorate a bit. Some, I believe, are parasitic. So as this one appeared to be growing on the ground on dead wood, I think it may not be parasitic, but rather saprotrophic. The very robust stinkhorn at the bottom, the mutinus, is probably different species from the more delicate ones that I've seen around here. As for the tiny earth stars on the right, they were abundant, especially under a hoop pine tree. There was also a whole garden bed full of the Phallus induciatus, looking like ballerinas on a stage. It's one of the most beautiful of all the stinkhorns, and it is more common in the tropics. The photo in the middle shows Phallinus noxious, which is a parasite and that causes brown root rot. It seldom produces a bracket like other Phallinus species. Usually this stocking or flat sheet with a white growing edge is all that you see on the outside of the affected tree. It's considered one of the more aggressively pathogenic of the parasitic fungi affecting large trees. And this one was on a native fig, which they fear is going to be doomed very soon. And of course, the lolly pink form of Marasmius hematocephalus is always eye-catching, and it was there in the gardens. We had quite a team of people joined us the next day when we went to Spiwa, which is in the Barron Gorge National Park at the back of Kuranda. Linda Reinhold, who's in the centre of that photo in the beige shirt, led us, and one of her favourite walks there. And though we didn't complete the whole walk, it was very wet underfoot, but we were finding lots of fungi. But nevertheless, quite the best find of the day was the male southern cassowary and his chick, who stopped us in our tracks and kept us entertained for several minutes before they simply melted into the forest. The little Panellus species on the left with the longish brown stem seems to be a different species from the ones that we see more commonly in southeast Queensland. And I guess the beautiful lavender pink coral is another species of Romeria, although I'm not sure. It seems slightly different in form. And I can report that the eagle-eyed Ori was expert in finding horsehair fungi that morning, and we found horsehair fungi of several different species. Davies Creek National Park is always a hotspot for fungi. I know I've seen different ones there every single time I've been. The Amaraderma, which we've called this thing Amaraderma rudae for many years, but Amaraderma genus was originally described from Guyana in the neotropics of South America, but it's now been split into a number of new genera and species, and the bleeding ones are now Sanguinoderma. And this 2020 study showed there are 10 different species of that genus, but all the Sanguinodermas are from our region, from Oceania and the Paleotropics. So it's now Sanguinoderma rudae. And some of these ones on that day were so wet as to appear almost blue, but they all looked the same underneath, and they bruised blood red when touched, and you can see that underneath the bottom on the left. There were also plenty of russulas at Davies Creek, including the blackening russula. It starts white, but any time it's touched, even the slightest touching, it turns black. It's a really disconcerting fungus, I found, because what looks beautiful to start with in a few minutes is grossly black. However, very excitingly for me, there was a new species of Marasmius. At first glance, it looked like a rather small Marasmius elegans with an extremely long and skinny stem. However, closer inspection showed that the stem was covered in reddish bristles. You can just see that in the lower right-hand corner photo. The cap also had short hairs. And I can only say, watch this space for this one. But also, please watch out for it and make collections if you possibly can. I don't know if it only occurs up north. It's possible many things that occur up north do also occur in southeast Queensland. So I would like to build up a picture of its variation and its distribution. We finally got to cross the Daintree River by ferry, of course, and entered the wild north. There is something different and quite special about the country beyond the Daintree. One important feature being that you're almost out of contact. Phones and internet seldom work, though I know Connie Kerr is hoping or trying to watch this Zoom tonight. And the one public phone I tried gobbled up my coins and failed to connect. However, the people we met were the same, generous, hospitable, and eager to share their treasures and local knowledge with like-minded folk. Connie Kerr took us to one of the blocks that's been bought back by a conservation group called Rainforest 4 to be conserved and eventually added to the World Heritage Cape Tribulation National Park. It was a beautiful area and had a walking track through it, which was fortunate for us because the forest there is often very dense. Lowland palm forest with black palms and fan palms scattered throughout. Matt was very excited to discover not one, but 2 new species of fan and spatula-shaped polypores growing on the aerial roots of this palm. I think that of the palms, the fan palm, Licuala ramsey, is one of the most beautiful of them all. Other green things included a couple of very strange cicadas which had been totally taken over by fungus, and they were attached to some trees. Diana Lehman's best guess is a metarhizium, but she said she hasn't seen it green like this before. And not from the Daintree, but from Macon's Beach in Cairns, although they do occur in the Daintree, was this large green white-lipped tree frog in full voiced, calling his mate, most of the night I might add, and it's the largest native frog in Australia. We were staying at Macon's Beach with Rhea Mayo, another member of QMS, and her hospitality was just fabulous. Further north along the Cape Tribulation Road, the state government has spent millions of dollars on a rainforest circuit walk that includes a boardwalk out into the mangroves. The Marja Botanical Walk was really magical. And apart from the sea snake that swam under us as we were walking the trail, and a pair of shining flycatchers that were nest building right beside the track, there were some fungi, including this polypore with the beautiful lavender pore surface. The pores actually stain orange when bruised, and although the colour is not showing very well, on the bottom right-hand one, there is some bruising, which should show orange. And Matt tells me it's called Physisporinus lavengelus. One day was really far too short a time to spend in the Cape Tribulation area, and Connie would love us to go back, or any others who are wanting to look at the fungi there. But we had to keep moving back across the Daintree River and west of Mossman to Gelatin. This is what a slide I've called sparse but special. These two agaricus species, the one we've already seen from Murray Falls and one at Gelatin, vastly different habitats. The one on the left is one of those that is a red staining agaricus, and Teresa was very excited to find that. She has another PhD student who is trying to sort out some of Australia's native agaricus. And these were beside the road in an old turkey mound, just right there along the roadway. And while we were in Gelatin, we met up with my main supervisor, Dr. John Durnley, who I have to tell you is a keen bird watcher. Maybe I'd call him a twitcher. He led us on the wettest day of the whole trip up Mount Lewis, quite a walk, to the site of a golden bowerbird's bower, one that hasn't been used for the last 3 years. This, the smallest of all the Australian bowerbirds, builds the largest bower. And it's formed into two what are called maypoles up to trees, and with a perch in between where the bird can display and court the female. I've taken the photo of the bird from the internet, and it was photographed by Laurie Ryan on Mount Lewis in 2016. I believe it was actually the same bower. So that is the bird that used to use that very bower where we were. The whole bower is constructed of twigs joined together with fungal threads. And they seem to be, from what we could see there, several different species, probably also different genera, all of which produce these rhizomorphs. The bowers are also decorated with lichens and seeds in the courting season. And these birds, according to the ornithologists, are threatened by climate change, and yet they're intimately connected with fungi. So it strikes me that there is potentially a big problem there, and how will this evolve over time? It requires a whole study on its own, and who's going to do that? There's a challenge for someone. We left Gelatin and went through to Malanda, and our last rainforest stop was in the Malanda Conservation Park. This tiny little park by the North Johnston River always delights. And this year, it was a huge display of Phylloboletus manipularis, which is the white fungus on the left, completely circling this tree. And also the soccer ball shape of a green ringtail possum peering at me from above our heads. And of course, another population of horsehair fungus. Then we relaxed for a couple of days with my cousins, Marion and John, at Topaz. And on one venture out in the rain, I found the first bleeding sterium that I've ever seen. I've heard of these from overseas on conifers, but have never actually seen one in Australia before. So I'd hoped that we would include a visit to one of the drier habitats on the western side of the Atherton tableland. And of course, in dry times, you wouldn't find a single fungus there. But with all the rain around, it seemed a perfect opportunity to check out the Herberton Range National Park. Unfortunately, we only had a short time there in Wongabel Forest, but it was rich in ectomycorrhizal fungi, boletes, russulas, and their relatives, including the beautiful multifurca on the right, Lacaria, hygrocybe, and more. I only found one marasmioid fungus, but it would be great to go back and spend more time there, as there could well be others. So, what are my conclusions from this trip? We had a great time, but after the foray, the real work begins. All the field IDs have to be checked. The specimens end up having to be lodged, and eventually new species have to be written up. I must say I couldn't have done the trip without assistance. The generous hospitality, the local guides and friends who encouraged me and shared the journey. A La Nina year has been perfect for fungi fruiting. We were so blessed with the weather. We missed cyclones and floods, but we got enough rain and the heat for the fungi to fruit. And there's plenty more to discover. So my encouragement is just get out there and start. Are there any questions or any comments from the audience here or online? Yes. Both ways.
How do we, Fran?
No, probably not. The question is ectomycorrhizal a term everyone understands? No, it's not. And fungi have a number of different roles. One is that they decompose dead material, vegetation. The other is that they partner with plants, and they partner with plants in various ways. But one of the important ways is through the roots of the plant, and they provide the plants with minerals and water, while the plant in exchange provides the fungus with carbohydrates, with sugars. And that can happen in a couple of different ways. Sometimes the fungus actually goes inside the roots. Sometimes it stays on the outside of the roots. Ecto means outside. And the ones that have that connection on the outside of the roots usually form fruit bodies, which we can see, big ones like the mushrooms that I was pointing out before. Other times, they actually go right inside the root, and those ones tend not to produce fruit bodies that you can see, although we did find the tiny little ones at Abergawi State Forest, which was their mostly their spores, which are enormous for those ones. So they're just two different ways of connecting, partnering with plants and exchanging nutrients. I'm asked, did I collect the stinkhorns? I'm sorry, I didn't. It was a very, very wet day, and collecting stinkhorns just wasn't really on our list. Did we see any bunya pines at Mount Lewis? It is their northern limit. Yes, I believe it is their northern limit. But I don't think we saw any. It was again, it was an incredibly wet day. We were sort of watching where our feet were going most of the time. John, did you see any bunyias? No. John isn't sure that he saw any that day either. Correct? Yeah.
Okay.
Why do some of the fungi bleed? Well, that's a very good question. I don't really know why they bleed. I mean, when they're bruised, they are obviously producing some sort of a chemical, but I have no idea why that occurs with some. All I know is that it's one of those features which you have to actually see in the field. If you wait until you've taken your specimen home and dried it, then that's gone. And it's gone forever. It's only something that you'll see in a fresh specimen. Yes. The question is, do you see any of the horsehair fungi down here? And I am able to report that you do. In fact, there are probably several different species down here. I was interested, we found a bird's nest on our property near Mullaney only about 3 months ago. And when I examined it, it was a spectacled monarch's nest, and it had a lot of the rhizomorphs incorporated in the building and the structure of the nest. And I thought, I've never seen any horsehair fungi on this property. This little bird is able to find it. I must be able to find it too. So after all the rain we had just recently, Bob and I went down into our rainforest, and probably less than 50 meters from where that bird had nested, Bob found, growing in the lawyer vines and tangling some dead leaves, horsehair fungi. We actually found two different varieties. And so one of them looked like Marasmius crinis-equi, the other one didn't. What else? Any other questions? Yes, Lil. So you found an agaricus on a turkey mound at Mount Cordo. Yeah, okay, that's good. Mount Cootha. Oh, at Mount Cootha, not Mount Cordo. Yeah, great. Any other questions? Yes. Distinctive smells. Are there any distinctive smells to the mushrooms we collected? The agaricus ones were very mildly smelling. Some agaricus are really strong smelling, but the ones that we collected had just a mild mushroomy smell. Of course, the stinkhorns all have a very strong malodorous smell, but part of the reason we didn't collect them. I just can't think of any others that have strong smell. The little Marasmius has no smell, I'm sorry. Do you have any idea how many species of rhizomorphic Marasmius you have found? Matt, that's a hard question. I don't, as far as Marasmius itself is concerned, this is looking at the genus strictly speaking, I have probably found only about 3 or so that I can, so far, that I've determined. But if I think of Marasmioid, that is things that look a bit like Marasmius, then I would say, a dozen or more. That's a conservative estimate. Any clues on the geastrum species of the tiny one? I do believe there is a geastrum minutum or something like that, but I'm not confident enough to determine that because I didn't take it and examine it. Are Marasmius species always gregarious? And that's a good question, because many of them are, and many of them will grow with different Marasmius species. So you'll find orange ones and red ones and white ones all growing together. There are a few that tend to be solitary, and just occasionally they'll have a whole family of them, but usually solitary. But as a rule, Marasmius do tend to be gregarious.
My initial concept of a horsehair fungus was those tiny fungi that have a little thin hair, like a horse hair on them. But now I understand that as a lot of Marasmius have that thin hair, and I thought that's what it meant.
The stem.
Was just a thin stem, but the term horsehair fungus actually relates to a different thing.
Yes, yeah. So Wayne was just saying that his initial idea of horsehair fungi was those that have a hair-like stem, and there are many Marasmius that have hair-like stems, and but that's not the concept of a horsehair fungus. A horsehair fungus actually has these bundles of hyphae, the fungal threads forming the rhizomorphs, and those threads can be meters long, and then the little fruit bodies can arise from them at right angles to the other threads. Yeah, Marasmius are I need to repeat that. What's an important function of Marasmius in the rainforest? Marasmius are generally saprotrophic. That means they are recyclers or decomposers, and they're really important decomposers of the leaf litter. Most of them, of course, are on the ground in the litter there. The horsehair fungus, Marasmius crinis-equi, has taken to the air, and it's very sneaky because it forms these litter traps, and it can catch leaves as they fall from the canopy and uses them up and decomposes them there in the air without the competition of all the other fungi that are on the ground. So it's a really neat strategy. No. Connie's asking if any specimens of the mutinous species that was seen at Cairns get collected. Not by us, I'm afraid. I don't know if Matt or any of the other people in Cairns have collected them. It may be they have, but we didn't. Did you learn of any interesting indigenous uses of the fungi you found? I did not have enough contact with the local Aboriginal people to learn anything about their usage of fungi. So unfortunately, we haven't advanced any further in that conversation. Perhaps in future we may. My feeling is that had I been wanting to study things that were of totemic significance or cultural significance to the people, they may have got back to me more quickly and made some asked questions about our research. But fungi are probably fairly low on the radar of the present day Aboriginal people, as they are on the radar of most Europeans.
Any more questions? Great. Well, thank you very much, Fran.
Thank you.